In the film Harry Potter, we can see Professor Snape at his very first moment of his life looking into Harry's eyes and says quietly, really have your mother's eyes. That line is so powerful, but not only because of the story behind it, because of the love he felt for Lily, but also because it touches one of the life's most ordinary yet magical truth, inheritance. So why don't we resemble our parents? I believe someone must have told you that you have your mother's eyes before, or you have your father's smile. So why do family features pass through generations and show up uniquely in each of us? The answer actually starts in the very first cells of our existence, in precise and beautifully random dance of biology. We will see processes of mitosis and meiosis, and I would like to help you walk through that dance of life and show how it makes each of us who we are. Okay, so hi everyone, I'm Jiaqi Chen, and I'm more than excited to be part of this BIO110's ambassador program and to lead you into cell division. I will first talk about mitosis in observing its stability, and I will talk about meiosis with the kind of physical model to see its diversity. And at last, I will talk about evolution and how it relates to our daily life. Every cell divides, but not every division leads to the same result. Every division can keep us alive, repairing wounds, replacing old cells, and also maintaining our bodies every day. But others make us different, shaping who we are and ultimately how life evolves. And today, I want to take you from what we observed in the lab to what we cannot directly see, to show how together they can tell us a bigger story, how life stays stable and how it changes. So first, I want to talk about mitosis, how we observe its stability. So in our BIO110 lab, we observed mitosis in a garlic root tip cells using a compound light microscope. Garlic root tips are extremely ideal for this experiment because they contain rapidly dividing cells. To prepare the sample, we treated the tissue with an acid to soften it, stained it with Gentian violet, and also gently squashed it into a slide to spread the cells into a thinner layer. But when I first looked through the microscope, what I saw was just not as clear as the textbook imagines. Some cells were blurry, some were unevenly stained, and at first I thought I did something wrong, but later I realized maybe that's how real biology observation works. Biology is not always clean or perfect. It requires interpretation, not just observation. Most of the cells we saw were actually in interphase, where chromosomes are not visible as distinct structures. This reflects something important. Cells spend most of their life preparing to divide, but not actually dividing. As cells enter prophase, chromosomes begin to condense and become visible as darker structures. In metaphase, we could identify chromosomes aligned at the center of the cell, forming what is called the metaphase plate. And the clearest stage we observed was actually anaphase, because in this stage we can see sister chromatids separated and move towards opposite poles. Because of this clear spatial separation, it was much easier to recognize. And this step is critical because even a step error in chromosome separation can lead to mutations or diseases such as cancer. Finally, in telophase, we observe the formation of two new nuclei as chromosomes begin to decondense. So even though the images were not perfect, we were able to identify the key stages of mitosis based on the chromosome organization. So we can see that mitosis is not just about copying, it's about precision and control. ensuring that each new cell receives exactly the same genetic information. And every time you can imagine your skin heals after a cut, your body replaces damaged cells. Mitosis is working silently in the background. But this actually raises a question. If life only copied itself perfectly, everything would remain the same. There will be no diversity, no adaptation, no evolution. So where does the differences come from? While mitosis is about copying, meiosis is about creating variation. To help visualize this elegant but complex process, I will use a simple physical model as a teaching aid. This hands-on demonstration, I believe, it will clearly show how chromosomes pair, align, and separate during different stages. Okay, so first let's look at these teaching aids. These are like small model chromosomes. For example, this red one represents the chromosome from the mother, and this blue one represents chromosome from the father. They're a pair because they're in the same size and shape, but they come from different parents. Now look closely. Each chromosome here is actually made up of two parts. These are called sister chromatids. This means that the DNA has already been copied or duplicated before meiosis begins. Also, there's one more important feature. At the bottom of each chromosome, here's one small piece that can be removed and replaced. For example, I can take this red piece out and then place one from the blue chromosomes. So this is designed to show crossing over when the homologous chromosomes exchange genetic material. This happens later in the tetrad stage, so we will use this function when we get to that part. So here's our starting point. A pair of homologous chromosomes, each already duplicated, with these two key features for the stages ahead. So first, chromosomes in the prophase of meiosis, chromosomes condense and become visible. Homologous chromosomes pair up to form tetrads. And this is when the crossing over occurs between non-sister chromatids, exchanging genetic material like I mentioned previously using this teaching aid. And in metaphase, tetrads mainly come from pairs of homologous chromosomes. They light up at the equator of the cell, attached to the spindle feathers, and this orientation is random, leading to independent assortment. And in anaphase one, we can see the homologous chromosomes are pulled apart to separate poles by the spindle fibers, and the sister chromatids remains attached to their centromeres. In telophase 1, chromosomes arrive at the poles, and the cell divides into two haploid daughter cells. Each chromosome still consists of two sister chromatids. And here we come to the meiosis 2, the equational division. There is no interface during this process. They just link to each other very closely. So in prophase 2, chromosomes still composed of two chromatids condense again, and new spindle fibers form in each haploid cell. In metaphase 2, chromosomes line up singly at the equator of each cell, similar to mitosis. And in anaphase 2, sister chromatids are finally separated and pulled to the opposite poles as individual chromosomes. And in telophase 2, which is the last step of meiosis, nuclear membranes reform and the two haploid cells divide again, producing a total of 4 unique haploid gametes. So what I show here is only 2 of them. So there are other 2 half. So what we can see from the key differences between the two stages of meiosis is that in meiosis 1, homologous chromosomes separate, reducing the chromosome number by half, whereas in mitosis, and similarly in meiosis 2, the sister chromatids separate, maintaining the chromosome number in the daughter cells. So this distinction is the cornerstone of their different biological purposes. Meiosis creates genetic diversity and produces haploid gametes. So for the reproduction, while mitosis produces genetically identical and diploid cells for growth and repair. So in short, without mitosis, there is no life. And without meiosis, there is no change. And this dance of cell division is very engine of life's continuity and diversity. So what if I told that you actually hold a backstage path to this performance? That your daily choices can actually influence its precision and its future? The answer actually lies in the precision of this process. Every time meiosis occurs, it's not just creating difference. It's assembling the genetic blueprint for a potential new life. And any error in this assembly, like, for example, a misstep in chromosome separation, could have profound consequences, leading to genetic conditions, and you can never imagine that. So here's the crucial part. Our choices influence its precision. Factors like smoking, excessive alcohol, or chronic poor diet don't just affect our own organs. So we can say that we have bad or an unhealthy lifestyle. It not only harm ourselves, it also harm others. Like they can increase the risk of errors in the very process that creates the next generation. So if we need to take that into consideration, we should take more responsibility in keeping a healthy lifestyle ourselves, which is not only better to us, but also better to the others. Because who knows? So understanding meiosis does more than explaining why we look different. It actually gives us a powerful reason to care for our health on a deeper level. It connects the dots between the apple you choose to eat and the decision to go for a run, and the fundamental blueprint in our future life. So when we live healthily, we're not just maintaining our own bodies, we are acting as responsible stewards of a genetic legacy. We're protecting the integrity of a process that has, against incredible odds, led to each unique one of us. So let's honor that together, not just for our own well-being, but for the boundless, diverse future of life itself. So last but not least, I would like to thank Professor Lee for encouraging me to overcome all the challenges in biology, as I have never chosen biology in high school before, and there used to be a lot of fear in my heart. And Professor Lee always show his honest feedback and inspire me to push through my limits. And I'm also really grateful to Professor Shuhua Li for the lab sessions, whose engaging and clear teaching mood actually made the concepts and all the complex topics really understandable, even for a non-biology major like me. And she always chat with me. Also, like Professor Lee, they also chat with us during the lab sessions to feel that they not only care about our academic progress, but also care for us as a whole person. And also, I really would like to thank my family and friends. I really want to thank you for supporting my path and my choice in choosing global health as a major. Although I haven't learned biology before, but I believe my academic journey in BIO 110 in this course, it has given me a solid foundation, and I really hope to use what I have learned in this class to help others to help more people in the future, to help everyone get a healthy life. And that's all. Thank you all. And I hope you enjoy watching my Ambassador program. Thank you.